All right. We got first couple chapters out of the way, and keep on keeping on. We've got the plasma membrane. I mentioned it briefly when we were talking about cell structure, cell physiology, that kind of thing, but it's so important in terms of what happens across it and the potentials that are set up on either side of it. It sort of governs everything to the point where we need to spend a whole entire chapter talking about it. So you're about ready to learn more than you ever wanted to know about the plasma membrane and how it works. Okay? All right. First of all, this is some stuff you should already know. In terms of, in general, you should know that the plasma membrane is you've heard this word, right? Phospholipid bilayer. So you've heard that before. What does that mean? We're going to look at what a phospholipid actually is. The bilayer comes from the fact, obviously, that there's two layers. You can see those here. Sort of looks like in this picture balloons with two little strings hanging off of them. They're pointing in opposite directions. They make this sort of sandwich in the middle. Now, what's important to note about the plasma membrane is as you look at it, there's a whole bunch of stuff all scattered throughout this plasma membrane. You've got some proteins, for example, they're just sort of embedded in the surface. They got stuff sticking off of them called glycoproteins. Glyco means what? Sugar. So a glycoprotein is like a protein with a sugar chain hanging off of it. Right? So glycoprotein. Glycolipid, you got this glyco chain, this sugar chain, but it's hooked to one of those lipids that are in the phospholipid bilayer as opposed to a protein. So you've got glycolipids. You've got phospholipids. You've got some other specialized membrane proteins. Notice this particular membrane protein spans all the way through from the outside to the inside. Okay, that's going to become important. You've got others that are differently shaped. It's like this one that spans the inside from the outside to the inside. You've got others that do the same thing. However, they are hollow in the middle. They're like channels that provides channel access from the outside to the inside. We're going to consider all of these things, especially these channels where things can pass through. Okay. Also, you'll see these little yellow dots on the screen, cholesterol molecules. Cholesterol is not always a bad thing, right? Hopefully you guys know that right now. There's different kinds of cholesterol, and even the two different kinds, well, there's actually more than two kinds, but probably all you learned about was the two kinds, LDL and HDL. Even both of those are necessary in the body. It's just, the proportions that they exist in is what's concerning. But you'll see as the semester goes on why cholesterol is important. When we get into hormones and we start talking about how hormones are made, you'll see that the precursor to all hormones is cholesterol. It's sort of the basis on which everything builds to make all the different types of hormones. So it's certainly important. Also, it's very important structural. I can't say that word. Structural. Yeah, it's very important for that reason as well. You'll see that in the lipid bilayer, for example, they're sort of spattered out for structural purposes and that kind of thing. So, okay. Obviously, we're dealing with basic structure here. The fact that it's like the way that it is, this membrane, gives the cell its shape. Now, it's flexible. It's very fluid. It can squish, and it can sort of distend a little bit. It can contract. It can swell. But ultimately, it's got some shape to it that it's held in with because of this phospholipid bilayer. It is important because you remember the hydrophilic, hydrophobic, and what that means. Philic means to have an affinity for, or to like, or to love. Phobic means to dislike or be afraid of, or whatever. Hydrophilic and hydrophobic, if we go back to this picture, realize what's happening, or what is present all out through here. This is the extracellular fluid, and this is the intracellular fluid, both of which is mostly comprised of water. So the hydrophilic ends are the ends that are chemically attracted to water, hence it's these heads that you see up here, and the ones that are pointing down that way. That leaves in between them this sort of space that's hydrophobic. In other words, it doesn't bond with water. If they bonded with water, if these tails were attracted to water, all of these would separate, and each one of these individually would stick with water. But the fact that they don't allows these to congregate like this, and only the heads be attracted to water molecules, while the tails are not, and by doing that, you create this boundary. You create this inside of a cell versus an outside of a cell, and that's very important. Okay, and we've got some fluidity. It allows the the cell shape to change a little bit, to squash and to sort of move around. And it also allows even these guys to shift in position with each other, sort of like it's a little sea of floating balls almost. So you can they can shift around and be fluid with one another. Now these guys can shift around even within that membrane, so it gives it a lot of what we call fluidity. Okay, like I said, some of these proteins are going to span the membrane as channels. For some pathophysiology, for example, cystic fibrosis. Cystic fibrosis is a condition caused by, it's a genetic condition, because somewhere in the genetic code causes the production of a faulty protein that is responsible for moving chloride ions in and out of the cell. Okay? So for example, it may be something like this, one of these channels that are specific for the chloride ion. In a person with cystic fibrosis, that's faulty. They don't control the movement of chloride in there as well. Like, for example, one of the tests that you do is a sweat test or a salt test to see how much actual salt is secreted by this person's sweat. The less that chloride ions, which is part of what you consider salt, right? Like sodium and chloride, those things make salts. The less that is regulated in the cell, the more it shows up, outside of the cells, like in perspiration and that kind of stuff. I don't know how much you know about it, and if you've ever read anything about it, but I was watching a documentary one time about a guy, one of the leading researchers out there in the field for cystic fibrosis right now, actually has the disease. And he remembers at a young age how he sort of figured out something was wrong was whenever he was active in sports and that kind of thing. And he would get really, really sweaty, and when he would get sweaty, he would hang his shirt out, just like, like thinks back to the locker room in football days. Smelling real nice in there. You pull your shirt out of the football locker and it's just standing straight up, He had a shirt, and his always had like actual salt crystals. He'd sweat. There was so much salt concentration in the sweat that he'd sweat out that there would be little crystallizations form on his shirt. So anyways, now you know that probably as well that one of the symptoms of cystic fibrosis, of course, is excessive mucus production in the lungs, and also, perhaps more importantly, at least in acute situations, in the pancreas. Mucus can build up in the pancreas as well and block those pancreatic ducts. And those enzymes and things like that can't be secreted as well, and they can build up and cause pancreatic damage and a lot of other things. But anyways, that's an example of something that can go wrong with channels in the plasma membrane. And there's probably a lot of other things that we could talk about. Other surface carrier molecules, they might not be hollow, but they can certainly latch onto something, and then that thing can sort of crawl down it to get from the outside to the inside of the cell, for example. Or it could grab it, take it in, flip around, and then open it, and then let it out on the inside. Just grab it from the outside and then turn and let it go on the inside, something like that. Those could be carrier molecules. Some are docking marker acceptors. That means that they're used for recognition. Okay? There's something about them that something else will come in, and some forms of communication will occur. They will dock. They will fit together, and cellular reactions could occur as a result of that. There could be enzymes. These are all some examples. You can read those yourself. Some examples of what these channel proteins or these membrane proteins could do and what they could be involved with. Cell adhesion molecules, CAMs. We'll talk about that a little bit later when we get to immunology. Things that allow cells to grab onto something sort of and stick, like white blood cells, for example. White blood cells have the ability to diapedese, which is go into those little cracks in your capillary and squeeze out of them and go out into your tissue, right? Well, you got to remember that the velocity of the blood is pretty significant when you're a cell. You're traveling through at a pretty high rate. How do these guys find these cracks, latch on, and then start wedging themselves in it? Well, sometimes they have these little molecules on the surface that are able to interact with other surfaces, like the capillary walls, for example. They can go and they can latch on with these molecules on the surface of their membranes and sort of hang there for a minute until they work their way in. Those would be like cell adhesion molecules. Okay. Take a look at a close-up view of what's called a phospholipid. Now, you hear a lot of terms as you go through your studies in the health field, one of which is lipids, which oftentimes are synonymous with fats. And then with that, you've got glyceride. You've got triglycerides. You think of fatty acids and saturated fats, unsaturated fats. Well, how do they all work together? How are they related? How do you make a difference in the distinction between them? Well, first of all, what a lipid is. A lipid is a component that has this right here. There's a glycerol, a little molecule of what's called glycerol. It's sort of like think of it as a mixture between like an alcohol molecule and a sugar molecule. Okay? Anything that ends in "all" is technically an alcohol. So it's some sort of sugar that's been converted into alcohol. Anyways, it doesn't matter. You've got a glycerol molecule right here. And to make it a lipid, what the glycerol molecule has the ability to do is have 3 chains hanging off of it to make a lipid. Here you see 2. These chains are called fatty acids. Okay? So to make a lipid, you've got a glycerol molecule and fatty acid chains hanging off of it. You can have up to 3, let's say, fatty acid chains hanging off of it. Guess what you call a glycerol with 3 fatty acid chains? Triglyceride. Okay? That's where the name comes from. Now, something about these fatty acid chains. What are they? If you could look at them and look at them chemically, what you would see is they are made of chains, called chains for a reason, of carbon atoms all stuck to each other in a line. Okay? So you've got this sort of zigzag line of carbon atoms attached to each other. And then on both sides of those carbon atoms, you've got hydrogen atoms attached to those carbon atoms. So it's just C's and H's. Now, there's a lot of energy in C's that are stuck together, and especially when there's only H's attached to them. If you don't believe me, then you don't believe that you get energy out of burning octane, for example. Octane that you put in your fuel that you put in your gas tank is a compound made of nothing but 8 C's with 10 H's around those 8 C's, okay, those 8 carbons. So, you've got a similar type of situation. You've got molecules here, fatty acid chains made of hydrogen and carbon. Now, why that matters to you? If this chain is holding as many hydrogens as it possibly can, if it's full, and all you've got is single bonded carbon atoms to one another, and then all the empty spaces are filled in with hydrogens, you've got what's called a saturated fatty acid chain. It's saturated, it's full, it's holding as many hydrogens as it can. If by some reason some of these hydrogens are gone, and instead of that carbon bond into a hydrogen, it forms a double bond instead with another carbon. Okay? So you've got carbons all in a row, and you've got hydrogens sticking out the side of them. If instead you started pulling off some of these hydrogens, and as a result, that carbon still wants to bond to something, so if it doesn't bond with that hydrogen, what it will do is make a double bond to the carbon right next to it. In that case, it is unsaturated. It doesn't contain as many possible hydrogens as it could. So that chemically is the difference between a saturated and an unsaturated fatty acid chain. Now, if you have those chains attached to a glycerol molecule, guess what? You've got saturated or unsaturated fats. Okay, then that's the difference between them. Anyways, if you've got this, a phospholipid. Remember, a lipid by definition has a glycerol group and it has fatty acid chains hanging off of them. A phospholipid will take away one of these 3 fatty acid chains and instead put on what's called a phosphate group. Okay? A phosphate group is made of phosphorus and oxygen, those elements. Now, you don't need to know that. But what you do need to know, and what you need to notice, is that these guys down here, they are charge neutral. They don't have any kind of charge associated with them, so they're not attracted to anything chemically. They don't want to, for example, they're not going to be attracted to water because they don't have that charge associated with them. A phosphate group does have a charge. This is why you have hydrophilic, hydrophobic regions on a phospholipid. You have a lipid where one of the fatty acid chains is replaced by a phosphate group, although it's on the other side. It's flipped up to the top. That's the head region. This is the tail region. The reason that we call this hydrophilic is because it is actually negatively charged and can be attracted to water. So it does attract itself to water. These guys don't. They form the hydrophobic regions. Okay? Now, when you have a bunch of these guys all together, you start creating a boundary for a cell. That's where all of this is coming from. Okay, now I don't expect you to know all the chemistry behind it, but hopefully you can at least follow what I just said to a certain degree. Okay, a little bit later we're going to start talking about in digestion For example, we'll discuss the Atkins diet. What's the bad things about it? How should it be used? Should it ever be used? What's the consequences of diabetes in terms of, how do these things play out? Like in terms of diabetes, at what role do they come into play? Acidosis, that kind of stuff. Why are ketones elevated? Like if my wife was a gestational diabetic, right? And, one of the classic things they monitor, for every checkup is the whole pee in the cup check for ketones. See if they're there. Why? What do ketones have anything to do with it? I'm sure some of you probably have administered, have had people in the places that you work that had to take this test. You ever stop and think about why you do the things that you do? Because I really want you to. I want you to know what you're doing and at least understand the basis of, if you're going to give someone a ketone test, why? Why are you doing it, and how does it be able to explain it to the patient in terms that they can understand? Not necessarily be the doctor and explain it to someone who knows science, but to explain it to someone who may not know what's going on, at least enough of it. And that's where we'll go with all of this stuff. So if all of what we're learning about doesn't seem like it applies directly to you, realize that at some level it does. At some level. This is the basis. You can't really understand everything that you do until you understand, in theory, how it all comes together. Okay. I'm going to leave these animations for you. I won't show those up here, but make sure you go to that website. Make sure you go through those animations on your own. Get an idea as to what I want you to focus on for this kind of stuff. Okay? It should be on the slides that you have. Yeah. Okay, here we go. Now, let's imagine that we have cells next to each other where their membranes are juxtaposed. I like that word. Positioned next to one another. Okay? Now, we have this extracellular matrix all throughout our body. It's sort of like this biological glue that keeps our tissues from falling apart. If there's two cells next to each other and they are forming a tissue, you can't just barely touch a tissue and it falls apart. I mean, there's something holding all of this together. What? Now, it's not like it's cement necessarily, but there is some, at a chemical level, something going on that is causing these cells to stay together. So they are held together by different means. Okay, there's three big, well, first of all, before I get into that, if you look up here, there's actually protein fibers are going to be involved with this, sort of like an overlapping and interspersing of protein fibers to help hold all of this together. You've got collagen, and you've heard of collagen, right? You think of collagen, you think of like the lip thing, right? But you've got collagen in your lips and your nose and your ears and a lot of other places. But realize that collagen itself is just a certain type of protein fiber that exists in the body. And what it's for is tensile strength. Tensile strength is sort of like pulling something this way. Like if I had a steel cable, it has a lot of tensile strength. I can't really budge it when I start moving in this direction. It supports a weight, like you're anchoring a big tower or something like that. You pull down that cable to hold it down. Elastin, stretch and recoil. You'll see this in your lungs, right? Being able to give a little, but yet to the point where it'll come back again. So some sort of flexible type of protein. And fibronectin, which is a little bit more solid in terms of structural, it's going to hold things together. Holds them in fixed position. Now, when these things come together, you see three different pictures up here of when two cells and their membranes are next to each other. How do they sort of anchor each other in place? Well, there's a couple of different ways. Let's look at, here's what's called a desmosome. Okay? Here's the three examples, and here's the pictures of each. Okay? This is called a desmosome. You know what a rivet is? Desmosomes are sort of like what's called a spot rivet or something. Something that sort of just shoots through and holds something together. If you'll notice here, these guys don't touch. What's nice about a desmosome, that's a place where, like a rivet like this, where two cells are held together. What they have are these protein filaments. Okay, a little place where the cytoplasm thickens and forms a little bit of a plaque, and these protein filaments that go through actually travel through this extracellular space into the next cell, and they anchor each other in. You can see how that's sort of anchored on both sides. But what's important about this is that there's a space between here. So tissue fluid, for example, can travel between these regions and between those cells. That's very important because that's very, very different, for example, than let's skip all the way over here to the other side. This is called a tight junction. This is almost like you take a sewing machine and you sew these plasma membranes together. You've got a lot of places where you've got proteins that are spanning the plasma membrane on both of these. They can come in contact with each other and form a tight chemical bond. And you've got a whole bunch of the proteins that do this all over the surface of that membrane, and it really tightens this up. Why is this important? Stuff doesn't pass through here. Well, what about your stomach? What about the lining of your stomach? You produce like gastric acids, right? You got hydrochloric acid in your stomach. You don't really want that stuff leaking between your cells and getting into your tissue fluid. So what you have on the lining of your stomach are a bunch of cells, for example, that are tightly held together in spaces where they're right next to each other to prevent like those hydrogen ions, for example, from the acid from going down in. Of course, you can feel the effects of this. Ulcers, of course, are when that mucous membrane layer and the cell layer is penetrated. And that's one of the reasons it hurts so bad and why it causes so much discomfort, because at that point, the acid can start getting into these spaces that were once tightly held together. Now there's exposure, okay, from those cells. Also, like in blood vessels, if you had this kind of stuff, realize blood vessels are made of cells, and the cells are anchored together to form a wall. It's not like it's just this tube that grows in your body. It's actually cells that are all stuck together in the shape of a tube. Well, realize that if every cell, as it's being stuck together, had a big old hole in it, you would have a perforated circulatory system, and you wouldn't be able to pump stuff very far. Okay? So you'll find that in the actual capillaries, or actually in the bigger vessels, these things are going to be sort of more tightly held together to keep stuff from leaking out. Now, when you get into capillaries, the permeability increases so it can deliver stuff to the tissues. Okay, and lastly, we've got gap junctions. Gap junctions are very important because it's like these tiny little shunts, these tiny little tubes that actually connect with one another. Each protein, there's one on each one, and they can lock up, then open the center, and create this direct pathway all throughout the surfaces of these membranes with adjacent cells, okay, right next to each other. This is going to be very, very important, for example, in cardiac tissue, whenever you have to rapidly spread out ions to conduct a nerve impulse from one cell to another cell. Instead of just limiting it to one little place, the more pores you have, the faster this stuff can travel through. Okay, you'll see that there's very, very little space. You can see the numbers 2 to 4 nanometers. A nanometer is 1 times 10 to the minus 9 meters, which means 1/billionth of a meter. Okay, so this is extremely small. That's 2 to 4 billionths of a meter space there. Over here, we've got a lot more. We've got about 20 billionths of a meter. So about 10 times bigger the space than what we're going to find over here. Here we have virtually no space at all. So these are very different, and they're going to be found in different parts of the body. Now, cells talk to each other. Okay, this is pretty weird to think about, but our cells do communicate with one another. Now, sometimes our cells receive information from people right next to them, just like us. We can receive information from the people sitting right next to us. Or we can receive information from a much higher source. Okay? And if you're into the spiritual realm, you can go from really high up, right? Well, our body sort of works the same way. There are certainly some chemicals, if you take a look here, for example, that don't go very far. They are meant for communication that is close by. Okay? One way that cells can communicate is through these gap junctions that I was talking about. And when I say communicate, realize that we're talking about chemicals. They're going to send chemicals back and forth to one another. And those chemicals, when they go from somewhere to somewhere else, when they get to their destination, they're going to cause a chemical reaction to occur. And that's cellular communication. Okay? This one cell may only release a chemical when certain situations exist. And that chemical can leave and go to another cell. And when it gets into this other cell, now something happens in response to that. Okay? A good example is if those of you who've taken micro, you learned about interferon. When a virus infects a cell will release a compound known as interferon. Now, interferon is a local messenger. Interferon will leave the cell that's now taken one for the team. That cell's going to die. But it will release interferon to neighboring cells, And that interferon will be taken up by those neighboring cells, and it will change the proteins being made to help guard against viral invasion for those neighboring cells. That's cellular communication. Okay? It's not conscious. They don't have tiny little brains in there. They're not working together. But it's a cause and effect relationship. Okay? Something happens, and there tends to be communication as a result of that. All right, one way is through gap junctions. Now, I realize that that's not drawn to scale. They're much closer together than that. Okay, but these gap junctions allow direct sharing of materials from one cell to another, especially in terms of like ions. Like say it's sodium ions, little charged atoms of sodium or chlorine or potassium. They can fly back and forth from one cell to another and cause things to happen through those gap junctions. You can have what's called a transient direct link-up of cells. The word transient means sort of fleeting. It happens every once in a while in time. They can come in contact with each other, and they might have receptors on their surfaces. And when they happen to click, boom, the chemistry changes, and it turns on a set of events inside each one of those cells. So that would be a transient link-up type of communication. Then we get into secretions here. You could have what's called paracrines. Okay, we'll talk more about this when we get into the hormone stuff. But paracrines are just a big generic group of things that are secreted by cells that don't go very far. They are secreted by some kind of a cell, and then it goes to cells that are close by. Okay, so what will happen is if you try to leave this, interferon, for example, would be an example of a paracrine. Interferon is released by the cell that's infected with the virus, and then it only spreads out to neighboring cells. When people figured this out and they realized what interferon could do, they thought for sure that it was a great cure and that you could use it to treat cancer, because cancer cells behave as if, sort of like what virally infected cells, how they would behave. There's a whole theory out there that cancer was originated as a viral disease, and now it's gotten to where it is now. They thought that if you could inject interferon with somebody, then you could stimulate all their cells to do this and fight off these viruses. Well, what they soon found out was that interferon is not stable in the bloodstream for very long at all. It deteriorates very quickly because it's not made for long-term hauling. It's made for very short-term distances. So paracrines are that. They don't go very far. They are secreted. They can bond to the surface, some of the proteins in the surface of a neighboring cell, and induce some sort of response in them. Neurotransmitters. You learned about this, at least, like acetylcholine and that kind of stuff, the stuff that crosses the synaptic gap at the very end of a neuron. Okay? That is certainly cellular communication. We'll get into that next chapter in detail. But these things that leave the end of a nerve cell float across this synaptic gap and cause something to happen in those cells, to pass that nervous signal on. That would be communication between cells, because both of those are types of cells. Then we've got what's called hormonal secretion and neurohormonal secretion. Hormonal secretion is you've got glands in your body. You know what your glands are, like your thyroid gland or your pituitary gland, or all this kind of stuff. The reason they're glands is because they are involved with the endocrine system. They're producing some sort of hormone. So they've got very active, like Golgi bodies, for example, because remember, Golgi bodies ship it, shape it, and pack it, and they're going to be excreted. They're going to go and be exocytosed by that cell and go somewhere else. That will be an example of something that's heavily going on in these types of cells. That's produced, these proteins or modified chemicals are produced. They're secreted by that cell. They actually go into the bloodstream. They're made for long distance. They get into the bloodstream and they travel. They could start all the way up here in your some gland up here, or it could start up here in your thyroid, and it could go to your big toe. It could go all the way down throughout the rest of your body. It could take a long path. They're made for that. Okay, these are hormones. Those are ways that cells communicate. This hormone, when it leaves, it could go to another cell way far away and induce a response in that cell. Neurohormones are actually, instead of being released by glandular tissue, by cells that make up glands, they're actually released by neurons. This happens in your brain. Okay, we'll talk about it a little bit later. It's kind of neat. You can have some nerve cells that actually release their neurotransmitter, but instead of propagating a nervous impulse, that neurotransmitter goes into the blood, and then it starts to act like a hormone, and it'll go somewhere. This is why you can do some things very quickly. For example, think about adrenaline and how fast you can respond to a fight or flight type of situation. and the sympathetic nervous system really kicks in, and you start breathing heavy, and your heart rate increases almost instantly. That's going to be an effect of this happening. This is going to be much quicker, right, than this, because this, you know how fast a nervous impulse can happen and release its product. This is a little bit slower and bulkier process than what you have here. All right, so I've already talked about that. All right, so we've said that cells can communicate, and we said that they can secrete something, it'll float over here, and it can induce some sort of response in the neighboring cell. That's fine and dandy. Now we got to explain why and how. How does it happen? How do you pass a message from one cell to another cell? And the more you know about this, the more you'll realize just how easy it is for things to go wrong. Okay, we have something called signal transduction. Fancy words. What it means is receiving a message and turning it into something meaningful. In other words, you can sort of think of it like, well, first of all, realize that the cell is very particular about what it's going to let inside of itself. There are certain proteins on the surface that are very specific to certain things, and it controls greatly what is allowed to enter the boundary of the cell. So if it can keep something from coming in, it will, as long as it can make something happen still. If you have a messenger, one of those things we talked about comes and it's knocking on your door and you're a cell. It will knock, it will ring the doorbell, whatever it is you want to think of, and what will probably never happen, well, not never happen, one thing that certainly is a possibility is that you let that have direct access and you let it just come in. You open the door and you let it come in. However, you're giving up some of your control when you do that. Another option that you have is to let somebody else answer the door for you, find out what that person wants, have that person, the person who answered the door, come tell you what it is, then you can tell that person or whatever, and you can use a middleman. Okay? You will see that sometimes our cells sort of do the same thing. Okay? And it'll do both of those situations depending upon what's going on. The butler, so to speak, who would answer the door and receive a message in your stead would be someone who is transducing that signal. The signal is whatever is being brought in from the outside. And by the time that is processed and the steps are finally brought to you, that's what we call signal transduction. Okay? Now, we've got, within signal transduction, we have what's called first and second messenger systems. This gets a little hairy, so try to stay with me here. Okay, a first messenger. Pretty basic. A first messenger, it says, produces an intercellular response by opening or closing specific channels in the membrane to regulate ion movement. Okay, easy enough. If some chemical floats in to the surface of this cell, knocks on the door, immediately what happens is that door opens and it comes in. It's that simple. Okay, that would be first messenger. The messenger is causing. Now, don't get me wrong. Let me clarify. That thing that is bringing the message is not actually what ends up entering. Okay, what actually happens is, for example, say this is a chloride channel. What it will do is come in, and there'll be a bonding site, for example, right next to that chloride channel. They could come in here, it could float, and it could attach to that bonding site. And when it does, it changes the chemistry on that channel enough to where now it opens, and chloride can come rushing in. Okay? So technically, the thing that came in to bring the signal is not what entered, but it caused something else to be able to enter. Okay? Now, you're giving up some control when that happens. You're opening it up and allowing movement normally to occur, diffusion, wherever it's going to take you. Okay? So you could, that's a first messenger. It produces that response by just opening or closing channels. Now, another thing that you can do is this first messenger could just pass the buck. The first messenger could transfer the signal to an intracellular chemical messenger. In other words, get to the membrane and then dish it off, give the message to something else that's in the membrane, and let that something else do the work. Okay, that's called the second messenger. You'll see what I mean when I show you this, when I show you the next picture. All right. Now, some things to keep in mind before I show you this picture here. We also have some channels that are not being controlled in our cell membrane. At the same time all this stuff is happening that we're talking about here, and we're exerting all this control, we've also got some channels that are called leak channels that are just open all the time. Crap can fly in and out of them. Now they're specific. Like in a chloride ion channel, potassium ion is not going to be flying through it. Only chloride ions will. The same for sodium, calcium, whatever. But you do have some that are relatively unregulated that allows just free movement. You've got others that are heavily regulated. It's very complicated. It's all happening at the same time. Okay. Now these gate channels are what can be open or closed based upon a stimulus. That's what I was talking about a second ago. It was closed. Messenger comes in, it can open. Now stuff that normally couldn't travel through is able to travel through that channel. Now, how do these things open, these gate channels? How can they be stimulated? Well, certainly chemically, they can be chemically stimulated by these things floating in and bonding to it and changing the chemistry and opening it. Other times they can be electrically stimulated. This becomes very important. I'll talk about this in a couple chapters. What will happen is an electrical impulse will travel through that plasma membrane. And as it's traveling through that plasma membrane, it hits certain proteins that are in that membrane, that are channel proteins, and they respond to that electrical charge. They were closed, for example. When they become electrically charged, now they're open. That's another way to do it. So you can respond to chemicals, you can respond to electrical changes, or you could also respond to just mechanical stretching. Okay, for example, the knee-jerk reflex. Okay, you guys have done that. You put it up, tap the patellar tendon, see if your leg goes up. Why does that work? You have what's called spindle fibers. I'll discuss this a little bit later in Chapter 8. Stretch receptors that are embedded within the muscle fibers that make up your thigh. And that is attached to that tendon that's just below your knee here. So that whenever you tap it, what you end up doing is, to a very small degree, is stretching the entire muscle. It's very, very small, the amount of stretching you're doing, obviously, but it's enough. You stretch it to the point where some of those fibers in that muscle are specialized to the point where their plasma membranes stretch a little bit because the cells stretch a little bit. And when they do, that stretching causes channels to open that normally would not open, and you get a nervous impulse sent. And that nervous impulse goes to your spinal cord, does a relay, pulls a U-turn, goes straight to your leg, and causes it to contract in fractions of a second. Okay? There's a lot going on there. We'll learn all about that a little bit later. Okay, now as far as the secondary messengers go, there are really only two that you have to know about in your body. There are others, but I want you to know about two. One of them is calcium. Calcium can actually be a second messenger. The other one is cyclic AMP. AMP, what's ATP stand for? Somebody tell me. Adenosine triphosphate. Right, so what do you think AMP stands for? What stands for, what's one? Mono. So adenosine monophosphate. Cyclic means it's used over and over. Okay, so this is cyclic AMP. Okay, what I want you to realize is, take a look here. This looks complicated, but it's not that bad. Because, let's take a look. Here's the plasma membrane, right? Here's one of the proteins that span the plasma membrane. It happens to be the receptor that we're looking at here. What can happen is a chemical messenger can come in, like you see here, and this is a first messenger. Now, if this was only a first messenger system and not a second messenger system, all that you would see is when this thing bonded with this protein, it would open and stuff could come rushing in and out. That was a first messenger system. The fact that it's what's called a second messenger system means the following. This would bond to that, and instead of opening, it changes the chemistry of this protein. When the chemistry of that protein changes, it's loosely connected to this little group of molecules right here, and since they're chemically connected, inadvertently, this chemistry changes. Okay? I'm skipping over a lot of steps, but that's all you need to know. This changes the chemistry of this. Because it changes, it's going to change the chemistry of that. You can surely follow the logic there. Okay? When this becomes excited and the chemistry changes, this alpha subunit, that's alpha, that's beta, that's gamma. If you don't know your lowercase Greek letters, it doesn't really matter. But the alpha subunit dissociates. It falls apart because it's absorbed more energy from this reaction that's happening. Now, when it falls apart, notice that it'll float around through that plasma membrane until it runs into a protein known as adenylyl cyclase. It's actually an enzyme. It ends in ACE, right? It's a protein that's going to be involved in a chemical reaction, so it's called an enzyme. When this adenylyl cyclase gets activated, normally it was not activated. Now it's activated. When it becomes activated, it will take ATP, so it takes energy to do this because you're going to burn ATP. Fortunately, we already learned how all of it was made, so we know where it's all coming from. You burn some ATP. It converts ATP into what's called cyclic AMP. So you basically dephosphorylate it. You pull off 2 phosphates. Now you've got AMP. That now becomes what's known as the second messenger. What you have done here is taken a message from the outside of the cell and brought it to the inside of the cell. Notice how complicated of a mess it was to make that happen. It's not as easy. There's no little runners that run back and forth that scream from one side to the other. You can't just open up and let everybody come in that wants to. It's got to be a very controlled process. You can see that through a series of reactions, you can start something happening just from the bonding of this all the way out here. So this is the first messenger. We call this the second messenger because this right now that it's activated, it wasn't activated before this response. Now that it's activated, what it can do is cause changes in the cell. For example, that activates something called protein kinase. You don't have to know these steps. Okay? That activates something called protein kinase A. What does that mean? Well, what that will do is protein kinase A is an enzyme that will phosphorylate, in other words, add a phosphate group to another particular protein that may be present in the cell. Well, what does that mean? Well, when you start messing with the shapes of proteins, you start messing with the ability for them to work. Okay? Either now you make something that wasn't working work, or vice versa. You make something that was working now not work. One of those two. So you control processes by doing that. Okay? Okay, so you alter the protein shape and function, and you get a cellular response as a result of that. Now, this is complicated. What do you need to get from all of this? You need to know the difference between a first and a second messenger system. And you need to know what two things, what the two most common second messengers are that are utilized by our body. What did I say they were? Do you remember? Calcium, cyclic AMP. Right. Okay, those are the two most common. Okay. Now, if you look at calcium, just so you know how it works, without going through all the steps, you get the same thing. You get some sort of interaction at the plasma membrane surface. The same alpha subunit gets activated, but instead of going to adenylyl cyclase, it activates a different protein molecule that's in your cell membrane. You get the same series of steps and conversions. Ultimately, what happens is calcium was stored up within different places inside your cell. Now it's released from those places inside your cell, and when it starts traveling around, it can cause cellular changes in responses. Okay, so that's why calcium is also considered a second messenger. All right. Easy, right? Clear. Got it? Questions about it before I go on? Okay. Where do you begin? Well, you know, review it. Make sure you read the part in the book. And if you have any questions, make sure you ask me. I know. Huh? Did you look at the changes I put on the website about the editions of the book? Did you look at that and see what pages I also told you to read? Okay. Check that. All right. Now, well, when I made all these notes, I was using the other edition of the book. 111 through 113. There you go. Check that. Okay. Why would we go through all the trouble of using a second messenger system? The biggest reason is because of what you see up here on the screen now. Amplification. You start with one thing, and by the time you're done, you get a huge yield out of what you were doing. For example, instead of having, what you got to realize is that we have tons and tons and tons of chemical reactions happening in our cells at any given time. And if we have to wait for one chemical messenger to come in and dock and create a response for every single one of those, we would get nowhere fast. So what we see happening in our cells is that, notice one of these bindings with that chemical messenger and that membrane protein can actually activate a lot of those adenylyl cyclases. Remember that alpha subunit falls off and goes over and activates that other protein, that adenylyl cyclase, it was what it was called in the case of cyclic AMP. It's not just one of those that gets activated. When that chemistry changes this protein, there's a lot of those little guys like this all around it, and a whole bunch of them get activated. And then each one of those produces a whole bunch of cyclic AMP, and it's just like a feed-forward process. It keeps getting bigger and amplifies at each stage, so that by the time that you're done, when you started with one combination, you've got a 10 million times fold increase in terms of what you get out of this versus what you started with. Okay? So, the product that you would get could be, you could get 10 million products, for example, on average, from just one interaction at the surface of the membrane. So you could see where it's a complicated step that's involved with this pathway, but you could see why it's so beneficial to us, because it allows us to mass produce this stuff and to actually get a whole lot more bang for our buck out of this. Okay. See how you know. Someone tell me what diffusion is. Oh, well, that's good. Okay, what does that mean?
You didn't ask.
Go ahead. Do what? Okay, why? So you're giving me a teleological definition. Okay, pressure is involved as well. I want you to make sure you understand something. What governs movement into and out of a cell more than anything else is what we call diffusion. Okay? It's free. Diffusion is free. It happens in nature. So I want you to understand why it happens, what causes it to happen. Why, for example, if I have 10 oxygen molecules in my hand, I have 10 O2s in my hand, and they're limited to my fist. Tell me why, when I open my hand, they spread out, and they will occupy the entire space of this room. Anybody know? You just know that it happens, and you just go through life not really thinking about it. The unexamined life is not worth living, folks. Let me ask you something. Look down at your desk. Is it moving? It is certainly moving. Are you moving right now? You're traveling 1,000 miles an hour, folks. Well, the earth spins, does it not? The sun comes up, the sun goes down. Unless you're still stuck back in Galileo's time, we revolve around the sun does not revolve around us. Okay? As you are spinning, the revolutions are what's going to make the seasons, but we're also spinning on the axis of the earth. That's the 24-hour period. Given the size of the earth and the fact that we make one complete turn in 24 hours, you can calculate how fast we are traveling, how fast we are spinning. We're spinning about 1,000 miles an hour right now. Does it feel like you're spinning 1,000 miles an hour? No. So when you're driving down the highway at 55, you're actually going 1,055 miles an hour. Assuming that you're going in the direction of the spin. If you're driving the other way, against the way the turn is going, you're going slower. Now, have you ever noticed, anybody fly? Have you gone on a long flight? Recently, a couple years ago, my wife and I flew to Taiwan. What is it, like I don't know how long it ended up being. 20-some, I don't know. It was a long flight. But it's faster one way than it is the other way. I don't know if you ever realize this, because when you're up off the surface of the earth and you're going from one point to another point and you're flying in the atmosphere, you got to realize the earth is spinning underneath you, right? So if the earth is spinning towards you, it's not going to take as long to get there. If the earth is spinning away from you, you're constantly chasing it and trying to make up time. Everything is moving. It's all relative. Okay? If you look at this desk right now, it looks like it's not moving to you, but if you zoom in and you look really close and you just keep like taking closer and closer shots until you get all the way down to the atoms, you will see that they're all just buzzing around like crazy. Okay? The reason that the table appears before you as if it's not moving is because these atoms that make up this table are not buzzing around each other, and they're not spreading out from one another. There are forces, chemical forces, that are holding them in close proximity to one another. However, what they are not doing is ceasing molecular motion. They are not just frozen and stopped like this. They're moving. It's just, it's sort of like if you can imagine, if I drew over here, if I drew a line across this board, and I drew it from here all the way over to there, and the whole time I was drawing it, I was making these tiny little squiggles in it the whole time. If you stood over here on the other side of the room, would you ever see those squiggles? There's no way. You would see a straight line, right? Sort of the same concept. You're looking at these atoms, they're all bouncing around and they're moving, but you're so far above them and so much bigger than them as you're looking at them, the resolution is not there. You can't see them moving around. It looks like they're not because the distance they are moving is so small. You know that you are mostly empty space, right? You look like you're solid, but in fact, you are mostly empty space. If you don't believe me, then you shouldn't be working in the field that you're working in. X-rays work, don't they? Computer imaging, all these techniques to produce pictures of people and films of their insides. This works. You're shooting radiation through people. And if it wasn't, like in the case of X-rays, passing through them, you wouldn't get these pictures. So how does it pass through people without just poking holes in them? It's because you're mostly empty space. What I like to tell my chemistry students is imagine this. The way to think of an atom is like you're at a giant football stadium. Say you're at you're watching the Colts play at the RCA Dome. Okay. Do they still call it the RCA Dome? I know they're building a new one. But, all right. Imagine a golf ball on the 50-yard line, right in the center of the 50-yard line. That's a nucleus. The rest of the stadium is the size of the atom, because the rest of the stadium is the space in which the electrons are buzzing around. It's mostly empty space. The actual mass of that atom is all concentrated into that little golf ball. So all of the weight that you can measure from that atom is actually in that little golf ball, and all these electrons are virtually massless, and they're flying around out here like crazy. Now that's freaky to think about, because all of a sudden, if you start looking down at the atomic level, you see that you're mostly empty space. Because your protons never touch, your nuclei of your atoms never touch, ever. Nothing ever touches anything else. Some of you won't buy that. Okay, you say, bull, why can I feel this? I can poke myself. I just got hit the other day. I know that things touch each other. No, they don't. Nothing ever touches anything. All we are is a temporal rearrangement of atoms in a way that are so close together as it appears that they are touching, but they are not. How do I know? Blow your mind. When I bounce a basketball on the ground, let's say this is the ground. Whoa, that's not the ground. Say this is the ground, okay? The ground is made ultimately of atoms, right? So let's say you've got atoms all over the ground. They're very close to each other. And around these atoms, you've got electrons buzzing all around. What do you know about electrons? What charge do they have? A negative charge. What do you know about two things that have the same charge? Are they attracted or repelled from one another? Okay, now keep that in mind. These atoms that make up the ground, everything that is the ground, their nuclei never touch, because the electrons are on the outside, and when the electrons get close to each other, they're all negatively charged, they actually push each other away. Okay? to the point where they never overlap and the nuclei come together. You can have chemical bonds, but that's way out of the scope of what we're going to talk about here. Okay? So, I bounce a basketball, okay, on this direction. It's coming down. It's approaching the ground. Realize that on the surface, this basketball is made of atoms, right? What's going to happen when these atoms come all the way down here and get really close to these atoms? It's going to push it apart. The basketball comes back up, right? Nothing has ever touched anything else. It never does in the universe.
What about when you drop something that doesn't bounce and it splatters everywhere?
Okay, you can keep track of that. You can certainly explain that. Because say it doesn't bounce. Okay, do what? Yeah, the movement changes, right? Like it splatters or whatever, and it breaks. All that you've done is spread the atoms out. Instead of going up, they went out. They went around. If something is now the softer this is, the more room that these atoms have to move around. Okay? So like if this is relatively soft, the ground's relatively soft. When they come together, the second they sort of start to meet, these guys can shift a lot of position. Now if it's a concrete floor and it's a glass that you're dropping on the concrete floor, you got to realize that these atoms are not going to be able. They're in a glass, for example, the atoms are more tightly stuck together. They don't have the flexibility. They don't have as much space between them as something like this that allows them to move around each other more. So when they come down, if that was a glass and you were getting here, what would happen is this is held together more strongly. If you drop it in grass, it probably won't break. You drop it on concrete, it will. Why? Because down here, in the case of like grass or soft dirt, these atoms are spread out more from one another and they're less fixated in their position. The harder the object, the more fixated the atoms are. They're still moving, but they're closer together. They got less give. So what happens is, if this is a hard floor and this is a glass, it comes down here, the force holding this together and pushing up in that direction is greater. And as a result, these atoms coming down here, they're not going to give, so instead they separate and they shatter and they pull out. They still haven't touched anything else. It's still a matter of keeping track of your forces. Okay? Take chemistry. It's good stuff. Yep, I teach Chem 111 in the summer. You're hoping I'd say no, so you could just say that and then say, oh, well, I can't take it, too bad. All right. Now, keeping that in mind, what I want you to remember now is that everything is in motion constantly. That's why diffusion happens. The fact that we have random motion. If I have oxygen atoms, I have them in my hand, and I open my hand, right? What's going to happen? Gases, things that are in gaseous form, the atoms are far apart from each other. That's what a gas is. If you want to make that gas a liquid, you cool it, right? And when you cool it, the atoms come closer together. They come closer together than they are in a gas, but not as close together as they are in a solid. We call that a liquid. If we cool it even more, we can get them to come even closer together, and we can get a solid out of that. So a gas, already, they're far apart from each other. So when you open up my hand, what's going to happen is they're going to now bounce around. They've got more room to bounce around. They're going to bounce around. And before long, they're going to bounce around and move to the point. They might collide with each other. They may collide with other atoms that are in the atmosphere of this room. But eventually, if you give them enough time, they're going to have random enough collisions to where they're going to have occupied the entire space of this room. They can bounce off that wall, fly over that way, fly up, fly down. They're going to take over, and they can expand all the way across. Now, to understand diffusion and what actually is happening, you've got to understand that concept. The reason that I can open up a pizza box right up here and that it can still be smelled way over there by Jody is because when I open it up, diffusion happens. You have a high concentration. They're all moving together right here, but the second you open it, you give them more freedom. That same movement is going to occur, but there's no restrictions on that movement. Nothing is keeping them pinned in. There's no collisions of a wall that's pinning them back in. So they can keep spreading out. If you were ping pong balls and this was like a lottery machine, okay, and I turned it on and we were all just bouncing off the walls, and the only exit was this door right here, you would see diffusion happen. And the reason is not because you're thinking, oh, I need to go out that door. Cells and molecules don't think like that. You can't say, there's the door, I'm going to walk out. What you do is you let yourself bounce around off these walls long enough until eventually people are going to find their way out of that. You're going to hit the right angle over a period of time. The more people that you have in here, the more likely you are to hit the right angle, someone, to get out that door. Now, let's suppose that that door led to another room, but it was closed, so that all we had was 2 rooms with a door in the middle. And let's say we all started in this room, okay? And we were ping pong balls and we're bouncing off the walls, and there's one door in the middle that goes to the other room. What's going to happen over a short period of time? What will you see happen? What do you think? Let's say we turned this on and I left it for 5 minutes or so. And you were going fast. You were bouncing off these walls. And I stopped, froze time after like 5 minutes, and I looked into these rooms. What would I see? Okay, but just generically speaking right now, you would see some in here and some in there. It's hard to say how many of each at this point, but you would certainly see some in here and some in there. Now, eventually, you're right. It would equal out. Why would it equal out? What does that mean? What do you mean by that? Do you mean that somehow consciously half of these guys are going to go over there, and then everyone's going to say, all right, we're good, stop, and then everybody stops moving? Do you think that's what happens? No. So what do you mean by it evens out? I'm trying to go with it here. Would you still, let me ask this. After those 5 minutes are up, and let's say that at first, if you were stepping out and you were an observer, and you were watching this happen, what you would see is you would see a mass movement in a short amount of time of a lot of the balls over here moving over to that room, right? You would see that. However, the more balls that accumulated in that room over there, what you would start to see is more balls coming back in this direction, right? You agree? Do you see why? The more increase of balls you get over there, the more you're increasing the chances to hit the right angle and for them to bounce back over here. So that's what's happening. This is where we're going to reach this thing called equilibrium, because eventually what happens is the amount of collisions over here by random movement causes them to hit that right angle and get through there. And the more, at first, what you see is a big change from here to all those things moving over there. But then at some point, as that keeps increasing over here, it starts to go back the other way. And what happens is you start increasing the number of collisions and the number of angles that can be met until you start reaching back over here. And then what happens is at some point, the amount of balls leaving there coming over here averages out and cancels out the amount of balls leaving over here to go over there. It's dynamic. It's not static. You still have changes. Everything is still moving. Nothing is frozen in time. You've got fluctuating movement, but it's balanced out. At first it was lopsided, so you saw a lot more move over there. And the more that you saw move over there, the more of those started to move back until eventually it sort of leveled out. Okay? All right. Now that you understand that, you can get a better idea of what diffusion is and how it works in the body. You set up these gradients like that. If you can set up a situation and load it, we'll call it loading. If instead of that door, that was a channel, a protein channel, and that wall was your cell membrane, guess what? It works the same way. If we can load up with potassium ions out here, and all of a sudden that gate opens, guess what's going to happen? Potassium ions can migrate that way until they start to balance each other out, and they're leaving sort of at the same rate, right? That's what's going to happen. Now, we're going to spend, as you will soon see, we're going to spend about 2/3 of our energy. That's a lot. We're going to spend about 2/3 of our energy keeping to make these pumping out sodium and potassium ions. You've heard of the sodium potassium pump, perhaps, in your other classes. We spend about 2/3 of our energy pumping up and making these gradients so that we have more on one side than on the other, so that they can be triggered and released at any given time and cause this movement to occur. It's very important. Okay, so consider this. We've got let's look at how this would apply specifically to the cell. Okay, here's diffusion. If we just had water here and we had something in here, say these actual blue things were water molecules, or say there's something else, it doesn't matter. There's something that we can see. If there's pores to travel through, or if it was wide open or whatever, you would see that, you've got movement in both directions. But what you will see if you start off with more on this side, at first when this thing starts, you will start seeing more of them start to form on this side until it balances itself out. Okay? So this is diffusion. Now what if you have something like this where you have the same setup, you have more on this side than you do on this side. So naturally what that means is that if everything was allowed to be the way it was, you would see diffusion in that direction at first, right? You would see diffusion happen this way. However, here's the problem. Notice that these guys will not fit in the membrane provided. There is a gradient there. There's what's called a potential. It wants to diffuse by law, by nature. It wants to diffuse in this direction, but it is physically prohibited from doing so. Keep that in mind. Okay, there's going to be a lot of places where diffusion becomes very important. Capillaries with exchange, carbon dioxide, oxygen exchange. In the respiratory system, up in the lungs, the same thing's happening there as it is in the body tissues. This is how oxygen, imagine this. You inhale a bunch of oxygen. So you have cells that are in your lungs that on the inside of your air sacs and the inside of those little alveoli, there's cells that line those alveoli, right? On the inside of those sacs, on that side of the membrane, there's a lot of oxygen. On the inside of I guess it would be the outside if you're thinking of the sac, okay? On one side of the membrane, there's a lot of oxygen that you just sucked in. On the other side of the plasma membrane, there's not a lot of oxygen, right? So guess where the oxygen wants to go based upon the principles that we've already talked about. Naturally, it wants to diffuse from a place where there's more of it to a place where there's less of it. Same thing with the body tissues. You produce carbon dioxide. We talked about the Krebs cycle. You saw where CO2 is produced in that cycle. We produce a lot of carbon dioxide. Guess what? There's a whole lot more carbon dioxide down in our tissues than there is in our lungs. So as the blood is traveling through our tissues, carbon dioxide naturally, because of its concentration gradient, diffuses in. And when it gets up to the lungs, it diffuses back out. Okay? It leaves and it comes back out. So you'll see this play. When we get into nephrology and into studying kidneys, that's a complicated mess. But when we get in there, you'll see how diffusion becomes extremely important. Okay, and moving these substances across these filtration membranes. All right, there's something called Fick's law of diffusion. What does Fick say about diffusion? What could possibly control, what could exert influence on how fast, for example, something diffuses? Because what you'll see is if you have a certain number of this and less of it over here and you let it go, and you have something else that's different, you have the same number of it and it's going from here to here, and you compare the two, you might see that one happens a lot faster than the other. Why would that happen? What could occur? Well, for one thing, probably the one that's heaviest is going to move the slowest, so that's going to probably affect diffusion. Things that are lighter tend to diffuse much more quickly than things that are heavier. That makes sense. The magnitude of the concentration gradient. For example, if I had 10 of us over here, and I had 5 of us over there, and I flipped the switch and we all started bouncing around. And at the same time, I had 5 of us over there and 105 of us over here and flipped the switch. Guess which one you're going to see happen faster? What diffusion is going to happen faster? The one where there's a bigger difference, right? 105 versus 5. That's called a concentration gradient. How high one end is compared to the other. Okay, so the magnitude, the numerical difference in the concentration gradient is going to make a difference. What about the plasma membrane that substances are crossing? Okay, what if we're having these objects pass through this door and go to the other side, but one of the objects is big and it sort of gets stuck in the door and it sort of has to get wedged through, whereas the other ones are really small and they can travel through very easily. You can see that diffusion would certainly be affected by that, right? How permeable is that membrane to the things being diffused across it? So that certainly would have an effect. The weight, I already talked about that. The distance through which it takes place. What if I had the same number, But instead, I narrowed our rooms down to only this big, right here on both sides. And then at the same time, I do the experiment and make the rooms this big. Whichever one you limit the space in is going to increase the rate of diffusion. It's more collisions, more chances to make it through. So the distance through which they have to travel is going to make a difference as well. Okay. Now, keep this in mind as well. Instead of just actual particles, you can also look at diffusion in terms of charge, electrical charge. This happens in our body. You can think of these things as objects, but in fact they're not. They're just charges. They're still atoms associated with these charges. But let's just look at it like that for a second. Notice that if you have a highly positively charged area, those positive charges are going to want to move towards a negatively charged area because there's less positive charges over there, and vice versa. You've got a highly negatively charged area. Those negative charges are going to want to move to a place where there's fewer negative charges. So you've got this sort of circuitry thing going on here. Okay. So tell me now, what is the difference between diffusion and osmosis? People say, I learned by osmosis, right? Put it up to your head. Is that osmosis? No. That's diffusion. It's not osmosis. So what's the difference? So you don't know. Anybody seen Osmosis Jones? The movie? Good movie. Good movie. You need to watch it. Anyways. Chris Rock does the voice of the white blood cell, the superhero white blood cell. Good stuff. Anyways, and then what is it? Drexenol, David Hyde Pierce, the guy off Frasier that was Niles off Frasier. He does the voice of the big robot, Drexenol or something. What his name was, Drex. I don't know. It was funny. Anyways, osmosis is diffusion, but it's the diffusion of water. Okay, so when we have diffusion of water, we have what's called osmosis. Okay. All right. Now, I want to end with this. I want to explain this, and then we'll quit. So stay with me here. If you have this situation right here, notice that we'll say this is 100% water. All these dots that you see represent water molecules. And you have this situation over here, well, you have the exact same number of total molecules present. See that? They're all the same in terms of number. However, what is different is that 90 of them are water, only 90, and the other 10 are made up of solutes, like salt dissolved or whatever, some type of other substance that's in that water. So how does this play out? What you're going to see happen is if you go back here, if I could join these two together and allow these things to cross, which way does water want to move? From left to right or from right to left? The water is going to want to go from here to here, right? It's a higher concentration of water over here. Not much, but it is. The solute wants to go from right to left. Okay, keeping that in mind. What if you had a membrane between those two, like we just said, that was permeable to both the water and the solute? In other words, they could both cross it. What you would see is exactly what you just told me. You would see water moving from the left to the right, and you would see that dissolved solute slowly making its way in concentration from the right to the left. Right? Correct? Yes. Okay. Now, so what you would see is ultimately this even distribution like you see right here. Okay. That's not so hard. What happens in this situation, though? Because this is what happens in our cells. What happens when you do have a membrane between these two, but that membrane controls very specifically what it allows past and what it doesn't allow pass? Okay? What if water can pass, the solute cannot? Will the water still pass? Sure it will. Will the solute pass? Obviously not. So how does that affect what you see? Look, the water can make it past. The solute is rejected. It doesn't make it pass. This is really interesting. Look at what we have here. You will actually see on the right side an increase in the volume of the water because the water molecules are still going to diffuse from the side with higher to the side with lower. The reason you have this additional is because you're not balancing this out with any other movement coming back the other way. Okay? So it loads up on one side. Think of it like this. What is happening? That salt, let's say it was salt that was dissolved in here. It's almost like, it looks like, and this is the misconception that people have, but it's not really true. It looks like that salt is drawing out the water. It looks like the salt is over here and sort of absorbing some of that water towards itself. It's drying out this side over here, right? Realize, though, that the salt has very little to do with it other than the fact that it can't cross the membrane. That's it. The reason for the difference in the loss of water and the gain of water over here is because of osmosis. It's a concentration gradient difference. Why do they limit your sodium intake if you have high blood pressure? Think about it. How many people do you know in the field that you work in that's an issue, right? You dispense meds or whatever, and you know enough people that come in to have high blood pressure, you know that one of the things they have to, you've probably told them now you need to cut out some of the salt in your diet or whatever. Why? Why would you do that? For this reason right here. This is what will increase your blood pressure. What if this was a blood vessel? This whole tube was a blood vessel. Okay? And you increased your salt intake and thus put more salt going through your body. Guess what would happen? Water, you would change the distribution of the concentrations of the dissolved solutes in the water to enough point where there's more salt on this side, right? There's less salt on this side. That means water is going to move in this direction. What happens when water moves into the blood? It gives the blood more volume, right? The more volume and the more blood that you have, the more pressure is going to be exerted on the containers, your vessels that are holding that blood. It's an osmotic gradient. When you salt your food, why does it dry it out? For the same reason. Okay? Right on the surface of those cells, all those juices that are coming out, you create this saltwater type of solution, and water leaves from the tissue fluid, from that meat, and comes up to the surface and actually goes towards the external salt solution that you are creating. That's why it's drying it out. Okay? All right, we'll start there.